All right, I'll read the question and then we'll go. Huntington's disease is autosomal dominant. That's very important. So I'm going to underline it, right? Using the pedigree, justify the genotype of individual H. In your answer, refer to the letters on the pedigree to identify individuals. Okay, so it's a pedigree question. We're asked to justify our genotype. Now, you could use a Punnett square. You can get away with not doing one, but I think you should anyways because it's good to justify genotypes with. So with Punnett squares, what are some things we usually need, right? We usually need a key, right? So like I need to assign letters to like the trait. So in this case, I might just use capital H is Huntington's, and then lowercase h is normal, right? Now, I could talk about the parents, but realistically, I only really need to say, look at this thing here, right? And look at their offspring. So, just by looking at the graph, right? Because I know Huntington's is autosomal dominant, this guy here has to be homozygous recessive, right? Because he doesn't have Huntington's at all, right? That leaves me with two options for this guy, right? He could be homozygous dominant or he could be a carrier, right? But I have to look at the children. I see that he had 2 kids, right? 4 kids. 2 of them don't have Huntington's and 2 of them do, right? And we know that whenever we have a homozygous or a pure breeding, I guess, Huntington's guy matching with a homozygous recessive individual, the only possible outcomes are heterozygous, right? But that's not the case here because we've got 2 not affected and 2 affected, right? Which tells me that the genotype has to be this here, right? Heterozygous. Now, what I should include in my answer, I'll just, because it's worth 3 marks, I'll just say, look, I won't actually write it out. You should include a Punnett square, right, of the heterozygous cross with the recessive. So H, 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 right? What you also need to do is, well, you need a key for the Punnett square. And you also need the genotype and phenotype. Right? After I've gotten all these things down, you could probably use some reasoning to explain why it has to be heterozygous and why he's not homozygous, right? So I might just fill in my Punnett square anyways, right? Right? So under reasoning to say why he is HH, I would just talk about the result of my Punnett square. Right? And then link this to the fact that J and L are unaffected. Right? But what I wanted to get at here is for this question, we don't have to use a Punnett square, but if you're going to use a Punnett square, you always should include, as like just good practice for any exam, include the key. So like this thing here, say, you know, capital H is Huntington's, lowercase h is normal. And you should always include the expected genotypic and phenotypic ratio, just as a habit, right? It's just a good habit to get into so that you don't like just randomly forget one and then lose a mark in the exam, right? So that's the first part. And then I said you have to reference the data provided in the stimulus, right? So I said here, look, I'm going to talk about the results of my Punnett square and link that to why J and L are unaffected, right? Which shows the marker I'm engaging with their stimulus here, and that should be 3 marks. Are we okay with that? Yes? Okay. This codon wheel I'll skip. You'll do it when you do protein synthesis. Now this question here, I'll just talk about it. Shh. Thanks, guys. All right. Just by looking at the graph, It's an explain question, but it's also worth 2 marks, right? So immediately, I know I don't have to like explain the whole process of Huntington's disease, but I should probably write enough about this graph to get 2 marks, right? So this graph just asks about the relationship between CAG repeats and age of onset of Huntington's. I think all you need to say is that as the number of CAG repeats increases, your age of onset gets lower, right? But that's just mentioning the trend. That's only 1 mark. You should probably reference numerically the data that the graph gives you, which should give you your 2nd mark as well. Does that make sense? Right? So identify the general trend, right? As CAG repeats goes up, as this goes up, the age of onset goes down, right? And I would probably just pull 2 data points just from anywhere in the graph to corroborate that statement. Right? And that should be enough for 2 marks because then you're explaining the relationship. Yeah, okay. I know I went really quickly. I'm going to spend more time on this question. So, diagram 1 shows a pedigree of a family known to be affected by Huntington's disease. Diagram 2 shows the gel electrophoresis on fragments of DNA from chromosome 4 known to be altered in Huntington's disease, right? Predict whether individuals S and U will be affected, and if so, at what age. Use data from the diagrams to justify your answer. Cool. So I look at this question. I see, okay, it's worth 3 marks, right? I have 2 pieces of stimulus I need to do, and the question asks me to look at if they'll be affected and what age, right? So I know some of us asked about, like, do I need to explain how gel electrophoresis worked? We don't need to do that, right? Because it's only a 3 marker. But you should show the marker. You can know how to read the diagram and interpret it, right? So first of all, I should like look at my diagram, right? So let's look at S and U, right? So S, he has around, I'll say 15 repeats, right? And he also lines up really well with P. Right, U is here. He has around, I don't know, 38, whatever. And he also lines up really well with Q, right? So we know that in gel electrophoresis, if 2 bands appear in line with each other, then they have the same length, right? Well, in this case, it tells us, right, the same number of CAG repeats. So all I do now is I have to look at diagram 1. Right? So in diagram 1, I see that P never got Huntington's disease, right? They were never affected, right? And that is also corroborated here, right? They never had age of onset, right? Which means that if S has the same number of CAG repeats as P, then S should also never get Huntington's disease, right? Because P never got it, right? That's the first thing I need to write down. The second thing is I look at U, right? So U, we don't know if he has it or not, but just remember, right? U is a child. So we know that Huntington's, the onset means like when they start to get it, right? And if U's a child, he probably isn't, he may or may not be old enough to get it or start getting it. So U is in line with Q, right? So they're both here. Now Q has Huntington's disease, but right now U doesn't have it, right? And the reason for that is, well, he's probably too young, because if you look here, the age of onset for Q was 45, which means that Q got Huntington's disease at 45 years old, right? And because he lines up with U, it's reasonable to assume that U also will get it at 45 years old as well, because they have the same number of CAG repeats. Does that make sense? Yeah? Okay, so for the 3 marks, I'll just say what I should write. Right, so I think to get 3 marks, you have to firstly identify that S equals P and U equals to Q from D2, which is diagram 2. Next, I need to say that P didn't get Huntington's. Therefore, S won't either. But Q did at 45, therefore U should as well, right? Right, so I'll explain what I wrote here. Right, so identify that S equals to P and U equals to Q, so that means they have the same number of CAG repeats as shown on diagram 2. That's why I wrote D2, which is what we see here. And then I also said we should also write about our conclusions, right? So because P didn't get Huntington's, which is what we saw in diagram 1 in the pedigree, therefore S won't either, right? But Q did get Huntington's at 45, which is data I pulled from my age of onset here and my pedigree here, right? Again, I'm referencing diagram 1 and diagram 2, and therefore you should get it as well at 45 too, right? And that should be enough for 3 marks. Cool? All right.